Go ahead and we'll continue with this stuff from chapter 12 here. So mitosis is going to be the actual dividing phase that alternates with interphase in the cell cycle. It spends most of its life in interphase. So that's why when you looked at those cells on the onion, most of them were in interphase. That is how the cell spends the majority of its life. So you're going to find most of the cells in there. When you look at interphase, it's actually divided up as well. M phase is what we call the mitotic phase. It's the shortest part of the cell life cycle in there. Interphase is 90% of the cell cycle. During interphase is when the cell is growing, it's going to copy its chromosomes, it prepares for division, but that's also when it's doing all of its normal cell type stuff in there. And we can divide that up into 3 phases in there. They're divided into 3 phases mostly because there are 3 major steps that they can be regulated along the way. The G1 phase is what we call the first gap. It's when the cell is growing. So it's a new cell, and it's usually going to start out smaller than full size. Here is where it's going to grow. The S phase is going to be the synthesis phase. Here is where it's going to make the chromosomes. It's going to grow some more in there. And then the G2 phase, the second gap, allows for more growth, and then you complete preparation for the mitotic phase in there. And then mitosis is broken down into 4 to 5 phases. It depends on which book you use. The textbook you have will break it down into 5 phases in there. The ones that break it down into 4 are going to... There is no pro-metaphase. That's the one that goes away, and it's split up into the other sections in there. So you've got prophase, pro-metaphase, metaphase, anaphase, and telophase that somehow disappeared off the notes. So with your mitotic spindle, this is going to be what stretches out across the cell to pull it apart in dividing. It's going to start to form in the cytoplasm during prophase. It's made up of the microtubules from your cytoskeleton and proteins, and what they're going to do is they're going to grow by incorporating more tubulin onto them to make them longer in there. When they shorten at the end, they just start to lose those little protein subunits. Your centrosome is going to be what organizes the microtubules and starts the assembly. And you've got centrioles that are located near the centrosome. We don't fully understand what centrioles do in there. We know that cells can divide without centrioles in there because they're not present in plant cells. However, we still are learning about these in there. So during interphase, a single centrosome is going to replicate to make 2 of them. And they stay near the nucleus. And then when you get to prophase, they start to move apart. And during the end of pro-metaphase, they continue moving. And by the end of it, they're going to be at opposite ends of the cell. There's going to be an aster where from the centrosome, it's going to make kind of a star-shaped area in there. And it's going to be a short array of microtubules that are going to spread out from the centrosome. The kinetochore is the proteins that are going to be associated with particular sections of the DNA in there that are going to face in opposite directions, and they're going to have some of the spindle microtubules attached to the kinetochores. So it's going to be the part of the DNA that attaches to the microtubules in there, which ends up pulling them apart. The metaphase plate is going to be the middle of the cell. It's halfway between the spindle poles. When you move the chromosomes out to the side, they're moved towards the spindle poles in there. And I know what I was going to grab when the skeleton sidetracked me. So when we look at it here, this is your mitotic spindle that's stretched across the cell. You can, if you look close, you can kind of see where you've got your centrioles here and what kind of looks like fibers coming out from it. That's your aster that does that. You can see them going out the other side too. The aster kind of blends in with the mitotic spindles in there. So the metaphase plate is going to be this midway point in here. And so at some point the chromosomes are lined up along the metaphase plate in there. So during anaphase, that's when the cohesins, which are the proteins that hold the chromatids together to form a chromosome, are going to be cleaved by an enzyme, and then they will separate into the two separate chromosomes. So we have some non-kinetochore microtubules that are going to be responsible for elongating the whole cell. So the non-kinetochore ones are not attached actually to the chromosome kinetochore in there. And they're going to be the ones that do this pulling and stretching apart on here. And then in telophase in there, you're going to have the nuclei reform during telophase in there. And then cytokinesis is generally going to begin in anaphase or telophase. So mitosis is where you divide the nucleus, is what that means. Cytokinesis is where you divide the cytoplasm. Generally, they go together. In human cells, they're going to go together, but there are a few organisms out there that will divide their nucleus multiple times and not the cytoplasm. And that's why this book separates it and emphasizes the separation, because it's a general biology for people going in all directions. In humans, what you're going to find occurs together. The thing that is probably the most studied of the things where they don't separate the cytoplasm in there is with malaria. It will have multiple divisions of the nucleus in there to make what we call a schizont. So in G2 interphase, in there, the nuclear envelope is bound to the nucleus, and you're going to have one or more nuclei. You're going to form centrosomes, and the chromosomes are going to form during the S phase, but they have not yet condensed in there. So when we move into prophase, in here, we have, I know this is kind of hard to see from a distance in there, but you're welcome to come up and look at the model at any point in there. Your chromatin is going to form chromosomes. So the little X-like thingies in here are your chromosomes. Your nucleoli is going to disappear. So part of it is here. It's starting to break down. That's going to go away altogether in there. Your sister chromatids are going to form. So they're going to form the X's where you have the two copies of the chromosomes come together and form the X. You're going to start to form the mitotic spindle. So that you can't see a whole lot in this one, but you can start to see it forming in here in this phase of the model in there. And your centrosomes are going to separate. So here they are close together at the top, the yellow pieces. They move a little bit farther apart. And now they're heading out towards those opposite poles on the cell. In prometaphase, here you're going to have the nuclear envelope in fragments. You're going to have the microtubules extend from each end, and then the chromosomes are more condensed. Some of the microtubules are going to start to attach to the kinetochore in the chromosomes and begin to pull it apart so that you can have the non-kinetochore microtubules stretching across as well. So this is showing kind of the middle of the prometaphase here. Metaphase is the longest of the stages, And these are going to slowly progress from one to the other. It isn't that instantly you go from one phase to the next. It is a transition, so it's possible when you look under the microscope you'll see cells that are in between these phases. Here your centrosomes are at the opposite poles in there. You're going to have the chromosomes aligned along the metaphase plate. That's the real characteristic thing out of metaphase, is they are lined up along this middle plate in there. And then the kinetochores of the sister chromatids are each attached to the kinetochore microtubules from the opposite poles. It's a really fancy way of saying these chromosomes are attached to that mitotic spindle. During anaphase, in here, it's the shortest phase. The cohesin protein is going to cleave the sister chromatids, so they're going to start to pull apart to become the separate individual chromosomes. They're going to start to move towards the opposite ends, and the kinetochore microtubules are going to start to shorten, and the cell is going to begin to elongate in there. So this is showing it progressing more. And more in here. So in telophase, here you have two new daughter cells formed. This is the end of the cycle. You're going to see them form two new nuclei. In there, you're going to have the chromosomes become less condensed. They're going to go back to being chromatin in there, and you have two new cells that get to start the whole process all over again. So with cytokinesis, the division of the cytoplasm, it's well underway by late telophase. In telophase, in a human cell, they're going to be pretty close to being synchronized, where you're going to have them pinching off. This pinching off is occurring in anaphase in a human cell, getting ready to divide. So during anaphase and telophase is usually when you'll see it in a human cell. What you're going to form in cytokinesis, which everybody seems to remember this because the term cleavage is in there. You have this cleavage furrow where it starts to pinch off in there. And you're going to have the appearance of a shallow groove that's going to occur in the cell surface near the old metaphase plate. So it is right in the middle in there. And you've got a contractile ring in there of actin filaments. It's the same stuff that's made up of muscles are made of. It's going to start to pinch off and close like a drawstring in there. So it's like taking and wrapping a string around the cell and tightening that string to pinch it off is what it's going to do. So the cell plate in plants, there is no cleavage furrow because they've got a hard cell wall. So the vesicles from the Golgi move along the microtubules, and they're going to start to form a new wall in the middle to divide things off. So that's going to be what most of our cells are going to do, but there are cells that are going to do some different types of replication in there. Binary fission is one of them that is going to go on in a lot of the single-celled eukaryotes. It's asexual reproduction. Bacteria do this a lot. They just split. There's no mitosis because they don't have a circular, or they don't have the chromosomes. Instead, what they have is a single circular DNA. They just make a copy of the DNA and they go on opposite ends of the cell. The DNA ends up attached into the cell wall to pull apart. The origin of replication is going to begin that division process. So where you've got these circular chromosomes in there, and you've got two copies of the two copies in there, it's double-stranded DNA. Your origin of replication is going to be where that chromosome starts to separate in there to make copies, and it ends up producing two origins. It's kind of like if you have two rings and you pull them apart, they're going to pull apart wider at the sides in there to spread directionally. So you've got your double-stranded DNA like this. If you start to split this open here with a bubble, you've got two origins that it can split. It can go down around this way, and it can go around this way. That's what we mean by two origins in there that are going to separate in there. And so your plasma membrane is going to start to grow inward after you've grown the extra chromosome to divide into 2 daughter cells. So evolution of mitosis, it's thought that mitosis had its origins in the prokaryotic binary fission. That's what came along first. And some of the proteins that are involved in the prokaryotic binary fission are actually related to eukaryotic cells doing mitosis in there. So we can see similarities in there. There have been possible intermediate stages in evolution, and they actually can be seen in something like the dinoflagellates. Also in diatoms and yeast. With dinoflagellates, they're going to replicate their chromosome and attach it to the nuclear envelope. And it separates as the nucleus elongates prior to dividing. So theirs separates before it divides. It's just a little bit different way of doing things. With the diatoms and yeast, the spindle within the nucleus separates the chromosomes. So when we do this division in a eukaryotic cell, the timing of it and the rate is going to depend. Some of your cells almost never divide. Some of them divide a lot in there. And so the cell cycle is driven by signaling molecules from the cytoplasm, and they tested this by fusing cells in different phases of the life cycle and putting them in with these different signals and watching what happened in there. So your cell cycle control systems are going to be cyclically operating molecules that are going to help to trigger the events, but it's also going to help to coordinate the events of cell division so that they can occur together in cells that they need to. And so it's going to have these checkpoints, which are control points where they're going to get their stop and go signals. That's where a lot of the naming came in for these G1, G2 phases in there, is because of where we found checkpoints in there. Animal cells are usually going to have built-in stop signals that have to be overridden. So by default, cell division will stop rather than just going on continuously in there. When they are overridden, when they shouldn't be, that can allow uncontrolled growth, which is what can happen in a tumor. Many checkpoints are going to come from surveillance mechanisms in the cell where they're looking to see what is going on in the cell and deciding should we divide or not. So the G1 phase has some checkpoints in there. They will call them restriction points in mammal cells. If it gets the go-ahead to progress through G1, then it's going to go ahead and usually progress all the way through the S phase, G2, and M phase. That's usually where it's going to be held up, is at G1. That's going to be the main one. If it does not get the go-ahead in there, it will go into what we call the GO phase or G0 phase, which is a non-dividing phase in there. Most of the human body cells are actually in the GO phase. They will mature. Things like your nerve cells and muscles are in GO phase. Occasionally, you can have those come out. Some can be called back out of the GO phase with external clues if you really need to. So your muscles, they can sometimes come out of those phases in there to help hypertrophy them. It's not going to be that just regular exercising all the time, normal day-to-day activities is going to call them out of that phase, though. I can tell I was drifting off to sleep when I was typing this. So your cell cycle clock, you've got the cyclins and cyclin-dependent kinases in there, those receptors that are going to be waiting for messages from the cell. The protein kinases are going to be enzymes that are going to either activate or inactivate by other proteins. So the kinases do phosphorylation in there. They're usually going to be present in constant concentrations in a growing cell, but they're in the inactive form, so they would have to be inactivated if you needed them to do something in the cell. So cyclin is going to attach to a kinase to activate it, and that's where you get the name the cyclin-dependent kinases that require a cyclin that will actively rise and falls with the concentration of the cyclin partner. So it's just the particular name of the protein that's going to regulate this cycle in there. MPF is the maturation promoting factor. It can be thought of as M-phase promoting factor in there. This one, when the cyclin synthesis is going to begin in late S phase through G2, it's going to be protected from being degraded, and it starts to accumulate in there. And it will combine with the recycled CDK, your cyclin-dependent kinases, and produce enough of the maturation promoting factor for the cells to pass the G2 checkpoint. So it's basically you're growing enough of this protein and enzyme that will come together that once you reach a certain threshold, your cell will say, okay, we've got enough of this, it's okay to move past the checkpoint in there. It's going to promote mitosis by phosphorylating various proteins. So we know it peaks during metaphase in there. The cyclin component is going to be degraded during anaphase. So you start to break down the amount of that regulator protein during anaphase. And then the cell will be terminating M phase, which it's almost done with during anaphase. It goes into telophase, and then you start the cycle over again. You have to start building up your materials to do the dividing. They get consumed in the process of dividing. Then the levels of it go down, and you start the cycle over again in there. So the MPF, it will act directly as a kinase and indirectly by inactivating other kinases in there. It's going to cause phosphorylation of various proteins in there in the nuclear lamina. Combines to the, or it's going to contribute to the chromosome starting to form the mitotic spindle in there in prophase. And then it's going to start to initiate its own destruction to lower the levels during anaphase. In animal cells, we have at least 3 different cyclin-dependent kinase proteins, and there are several different cyclins at the G1 checkpoint. More what you want to understand is that there's these external factors out there that are going to be controlling the regulation of when a cell divides. These are still relatively new because right now we're saying we have at least 3 different ones of these. These are things that we have learned about in the last 10 years. So it's relatively new. There's going to be more of them found in all likelihood that aren't even named yet in there. So rather than saying you need to know specific names of which protein goes with what here, the overall concept that there are these proteins in there that are going to control the enzymes and regulate when a cell divides, that's going to be more useful to your understanding than those details in there, which is part of the reason for making this one an open note test, because I just don't see the point in saying memorize all of those names when you're not likely going to use them. So there are stop and go signals that are going to be at various other checkpoints in there. You've got internal signals at M phase. When Anaphase has the separation of sister chromatids, part of that is going to be regulated. It's not going to begin until everybody lines up along the metaphase plate. So one of the reasons for having a checkpoint in there is if you don't have everybody lined up and you start division, you leave people behind. You don't want to leave chromosomes behind in a cell. the cell will generally not survive. So that's one of the important reasons for having these checkpoint regulations, is you can make sure everybody is behind. And it's kind of like if you've got, I had a friend growing up, and her family had 15 kids, and they would go to church in their school bus that they had. And her mom would always tell stories of, oh, I came home and I left 2 of them behind. They needed to have a check system to count and say, we have this many children on the bus so that we don't leave any behind. That's essentially what this is doing, is making sure you're not leaving any of your kids behind in there. Growth factor will be released by certain cells. It's going to stimulate other cells to divide. These are studied a lot with cancers in there because they're trying to understand what makes certain cells start to grow out of control. So far, we've discovered over 50 growth factors in there. And so we have just opened that can of worms in there. One that's been studied a bit is the platelet-derived growth factor in there made by platelets. So when you have an injury in there, this is going to be required for division of the fibroblasts that make the fibrins in your clots in there. So when you have injury, these will proliferate to help you heal during that point. Some of them are going to look at density. So you have density-dependent inhibition. This will prevent cells from overcrowding an area. Some cells are going to have anchorage dependence. Most animal cells need to be attached to something to divide. They can't just be floating around randomly and dividing. That's one of the things that goes wrong in cancer cells, is they don't necessarily have this anchorage dependence in there. That's what allows them to float to other areas and do metastasis or start new tumors. Most of your cells, if they aren't attached to something in a proper place to grow, you can't start dividing until that happens. So you have a loss of cell cycle controls in your cancer cells, and that's why the amount of knowledge in this area has increased in the last 10 years, is largely due to cancer research. So cancer cells do not follow the normal signals regulating the cell cycle. They no longer follow the rules. They're doing things their own way in there. They lack density-dependent inhibition, so when the area is full, they don't care, they just keep on dividing. They lack the anchorage dependence, so they can freely float and plant their seeds other places. If and when they stop dividing, they'll do so at random points. So normally a cell is going to stop, if it's not going to divide anymore, it's going to go into the GO phase. Cancer cells stop wherever they feel like it in there. Where we ended up with a bunch of these cells was the HeLa cells that came from this woman, Henrietta Lacks, in 1951. She had a cervical cancer tumor that was removed, and it's kind of a weird set of coincidences that led to us having these cells, and they were collected, and there was a researcher who was trying to be able to culture human cells. And most human cells, you can grow them for about 50 cycles, and then they're going to stop and die. They will just kind of age out. They've reached the end of their dividing cycle. And he had collected these from her tumor, and these just kept on dividing. So they were a big deal because they were the first immortal cells in there. And they were spread around to different researchers in there. He had them, and it was before you had patents being on human tissues and everything, so they ended up being sent to other labs through other researchers. He would contact them and say, yeah, I'll send you some of these immortal cells so you can start studying them. And they eventually were spread all over the place. There was labs that took over and made them as factories in there. A lot of other cells ended up being contaminated by these in there. It's really kind of an interesting story how these cells ended up being spread all over the place. And so you'll see them, they use the first 2 letters of her first name and last name in there. HeLa is how they got their name. They're used for a lot of research things because they do replicate over and over again. You're not limited to those 20 to 50 cycles in there. So when a cell has changed where it converts to a cancer cell, we say that it's been transformed. Normally in the body, an immune cell is going to destroy this, but if it evades destructions and proliferates, it can become a tumor. And some tumors can become cancerous. So the tumor is just a mass of abnormal cells in otherwise normally functioning tissue. A benign tumor, the abnormal cells remain at their original site. They're not moving around. And they can generally be completely removed by surgery, and that's usually the end of what you have to do for it. If you're going to have a tumor, that's the kind you want to have. The malignant tumor becomes invasive enough. They can start to impair functions of one or more organs in there. A lot of times they impair function simply by their size, and they will restrict nutrients to other things, and they just occupy space that that other organ needs. Malignant tumor cells are going to be abnormal in a lot of ways. They may have an unusual number of chromosomes in there. Their metabolism may be disabled. They are really self-serving. They generally have no useful function to the organism they live in. They can change their surface so that they can start to lose attachments, and that's what allows them to be able to spread throughout the body in there. And they can secrete signal molecules for angiogenesis, which is developing blood vessels. So when they go and they float around to some other place, they'll actually send out the signal to say, you know what, body, create a blood supply for me here, and they will do that. So some of the chemotherapy drugs will actually work to prevent that angiogenesis, to try and starve a tumor in there. So metastasis is when we have separation from the original tumor and it proliferates in a distant location. Generally, metastasis is not a good diagnosis to have in there. So you usually have to do chemotherapy that's going to be more diffuse in the body, because you can't predict everywhere where one of those cells is gone. Once they know one has gone, one of the reasons they check lymph nodes, if one has broken free, odds are other cells have broken free as well in there. So how we treat cancer right now: high energy radiation. This is helpful on a localized tumor that's staying in one place. It will damage the DNA in the cells, the cancer cells, more than the normal cells. And so the majority of the cancer cells then lose their ability to repair the damage and die. And also at the same time, hopefully your immune system is trying to overcome and take over some of those cells. With chemotherapy, these drugs are toxic to actively dividing cells. So that means they're going to be toxic to your other cells as well, but cancer cells tend to divide quicker than other cells. So the hope is that the cancer cell will eat up more of the toxin and die before the whole organism dies in there. So these will be transported through the circulatory system. There are a few that are oral. Most of them are delivered through IVs in there. And they're used to treat suspected or known metastasis in there. One of the drugs that's been around for a while, Taxol, is going to freeze the mitotic spindle. It prevents the microtubules from depolymerizing. So part of that separating is when they start to break back down. And it stops that from happening in there, so they can't go past metaphase in there. So that's given to some patients after they've had cancer to try and prevent it from coming back in there. All of these drugs are going to have side effects due to their damage on normal cells. So there is no cancer treatment that does not have side effects. And realistically, to say that we would have a way of treating cancer without side effects, with what we know about dividing the cell right now, it's not looking like that kind of treatment is going to be developed in there. If this stuff with the cancer is interesting to you, there is a video that Nova put out. It's been around for a while, but it talks about the angiogenesis inhibitors in there. It's one of those, I think it's about an hour long. And then there's the book, The Immortal Life of Henrietta Lacks, that gives the history on the HeLa cells, that talks about those things if either of those are of interest to you in there for some point in the future. Is there any questions on this part?